Welcome, and thank you for joining this preview of our work at Generation Bio ahead of our public offering. I'm Jeff McDonough, CEO of Generation Bio. I'm really excited to share our vision around creating a new class of gene therapy, durable, redosable, and scalable for millions of patients living with rare and prevalent diseases. First-generation gene therapy has made strides towards durability, but true redosing and scale are unprecedented features. And as I said, I'm very excited to walk you through what has led us to be able to offer these features and where we believe it will take us as a company. In this presentation, I'm going to give you a comprehensive overview and introduction to the company, walk you through in detail the scientific elements of our platform, and introduce you to our portfolio before summarizing the events that we see unfolding on our journey towards building a major biotech innovator. The ambition to create a new class of non-viral gene therapy requires a unique combination of breakthrough technologies in construct, delivery, and manufacturing, integrated to create a proprietary and highly disruptive platform. All elements of our platform are capsid-free, yielding a short 8-week research cycle and truly large-scale manufacturing, which combine to yield the rapid development and scaling engine that's at the heart of Generation Bio and allows us to address rare indications and previously unreachable prevalent ones. This leads to our broad and expansive portfolio of 8 wholly owned programs across multiple tissues, 5 in the liver, 3 in the retina, with 3 well-defined expansion areas. We've assembled a world-class team and group of investors to support our work here at the company. The team shares over 40 IND filings and 20 approved products in our history, and we've assembled a terrific group of long-term healthcare investors through 3 rounds of financing, most recently closing a round of $111 million in January of 2020 in a Series C round. We hope you'll consider joining our team for the next phase of our journey as a company and as a community. Generation Bio is entering a landscape of significant achievements in genetic medicine. AAV has contributed durable efficacy to the field, but also comes with significant constraints. It excludes many patients, is a single dose, and has significant constraints in both the scale and costs of manufacturing. Gene editing shares a very similar profile. mRNA has brought redosing and scale to the table, but is constrained by relatively short half-life and a limited application to chronic diseases. Nevertheless, these modalities have created significant equity market value, and there's lots to play for here. What if you could combine the durability of AAV with the scale and redosability of mRNA? We aim to unlock the full potential of gene therapy, durable, redosable, titratable delivery of large genetic payloads to new tissues at scale, offers a game-changing set of characteristics for the key stakeholders in this field. For patients, it offers the opportunity to titrate to target expression level for each individual, to begin dosing in childhood before disease progression has begun, and to redose to extend benefit over a lifetime. Physicians gain the ability to address new indications with larger and multiple genes, to rescue previously undertreated AAV patients, and to extend gene therapy for the first time to millions of patients. For payers, the ability to better predict clinical outcome and to know that redosing can ensure durability of benefit can allow them to reimburse in the current paradigm, a feature we believe will be crucial for adoption and ultimately for disruption. Our proprietary non-viral gene therapy platform is composed of 3 core elements. We begin with a novel construct called closed-ended DNA, or CEDNA. This is a fully double-stranded, covalently closed-ended DNA construct which yields durable expression of large genetic payloads in the nucleus. We have invented the first-ever cell-targeted lipid nanoparticle delivery system that enables redosable delivery to multiple tissues. And we've invented a capsid-free manufacturing process that functions on standard biologics infrastructure to manufacture our constructs with scale sufficient to support millions of patients. We have established a leadership position in the non-viral gene therapy space with 36 independent patent families filed to date. Our broad and expansive clinical portfolio consists of 8 wholly owned programs, 5 in the liver, 3 in the retina, with 3 expansion opportunity areas in skeletal muscle, oncology, and the central nervous system. Across this broad portfolio, we've selected indications with well-established endpoints and a clear path to human proof of concept. We've also paid attention to the interconnectedness of the pathobiology across indications within a tissue. PKU, for example, is an intracellular disorder in the liver. Success there leads in turn to diseases with similar pathobiology like Wilson disease. In hemophilia A, we seek to use the liver as a biofactory to produce a therapeutic protein, and this approach can be applied also to Gaucher disease and to antibody gene therapy. We are pursuing a similar deliberate approach in the retina, beginning with a rare indication in LCA10 and progressing through more prevalent indications like Stargardt and wet AMD. Our strategy is to rapidly expand within each tissue area following human proof of concepts. We are working in parallel across a series of tissues: liver, retina, muscle. I'll show data on each of these tissues in today's presentation, as well as in oncology and CNS. This deliberate progression leads to the advancement of a rapid human proof of concept indication in each tissue. I take here in the slide the example of secreted proteins from the liver and our lead indication within this application of hemophilia A. We can use our 8-week research cycle to rapidly pre-fill a pipeline of both monogenic indications like, for example, Gaucher disease, Pompe disease, and HAE, as well as in parallel highly prevalent indications such as neutralizing antibodies against CoV-2 or HIV. Neutralizing antibodies today typically deliver between 3 to 6 months of efficacy per dose. On our platform, we aim to deliver the genetic sequence for these antibodies, allowing the patient to produce the antibody from their own liver, yielding the potential for years of efficacy per dose. We have invested from our founding in a proprietary capsid-free manufacturing methodology, which now has established capacity to support our broad and expansive clinical pipeline. We have scaled our process to 200 liters, which at a current 3-week time in the plant offers efficiency and yields typical of biologic processes, which is unprecedented in the gene therapy field. We have matured this process to cGMP readiness, and we have tech transferred this to a third-party GMP provider who is routinely running our process at 200-liter scale. In addition, we are underway with tech transfer to a second contract manufacturing organization where we have access to 4 additional cGMP suites. This existing capacity gives us clear line of sight as we scale to larger upstream capacities to create hundreds of thousands and ultimately millions of doses a year. This scale and progression to a variety of redundant and progressively larger providers allows us to create cost of goods that are close today to many biologics. And as we continue to scale, we believe our cost of goods can be equal to and even superior to many biologics in the future. This combination of long-term efficacy and progressive operating leverage and manufacturing scale supports our vision to move beyond rare diseases to support highly prevalent diseases in the future. I'd like to turn now to a deep dive on the platform science behind Generation Bio. The structure of ceDNA imparts key features to support the characteristics of a non-viral gene therapy platform. It is defined by 3 elements structurally: inverted terminal repeats at either end of the molecule, which are typical of those found in AAV. Linear, double-stranded, intermediate DNA structure, which is covalently closed-ended. This structure enables two key features: access to the nucleus and non-integrating episomal expression. The first of these features, access to the nucleus, is demonstrated on the lower left of this slide. In this experiment, we show that when plasmid is injected to the cytoplasm of the cell containing a gene for green fluorescent protein, that protein is not expressed, indicating that plasmid DNA does not translocate from the cytoplasm to the nucleus. When inverted terminal repeat structure sequences are integrated to plasmid, you see a small increase in the amount of GFP that is expressed, intimating that these structures are important for translocation to the nucleus. Lastly, when the fully linear covalently closed-ended structure of sedna is injected to the cytoplasm, you see a dramatic increase in the expression of GFP, again, inferring that we are penetrating the nucleus in order to set up that expression. The second feature, non-integrating expression, is consistent with the profile shown in the lower right. In the left of those graphs, when sedna is introduced in vitro to post-mitotic cells, you see an increasing and durable expression of GFP. In contrast, in rapidly dividing cells shown at the far right, you can see dilution of expression consistent with episomal non-integrating expression. Moving to in vivo proof points, here we show durable expression after a single IV dose of sedna encapsulated in an LNP. At left, we're using an intracellular reporter protein called luciferase, showing stable expression for 6 months after a single dose. At right is a secreted human factor IX protein, showing stable expression over 2.5 months, similarly after a single injection. This sets up the key experiment we show on the next slide. This slide outlines the experiment that first-generation viral gene therapy cannot do: the ability to demonstrate redosing and expression at a higher level in immunocompetent mice. Again, with the same two reporter proteins, we show the ability to stack expression now on top of existing levels by redosing about a month out from the original dose. On the left, a single dose of luciferase is followed by a redose at about 30 days, showing an increase in expression, and at right, factor IX shows the same kinetics with the same redosing. This durability and lack of immunogenicity upon delivery, which is derived from our non-viral delivery LNP platform, translates to a key clinical feature we call titration. This allows for each patient to be titrated through multiple doses as needed to reach an individualized therapeutic goal. sedna also has the unique capacity to deliver large genetic payloads. We have routinely created closed-end DNA constructs up to 12 kilobases, roughly 3 times the capacity of what's possible with first-generation AAV gene therapy today. And we can use that genetic payload capacity to create a variety of new opportunities in gene therapy, beginning by improving expression for existing gene therapy targets through more efficient or more potent construct designs that take advantage of this larger payload capacity. We can incorporate genes that are too large to be packaged in viral vectors, which are constrained by the size of the capsid, unlocking access to new genetic targets and diseases. We can encode for proteins or transcripts to enable multiple therapeutic mechanisms, including combined knockdown and replacement of full genes. And finally, we can use this capacity to add native or engineered element for context-dependent regulation of expression. I won't go into further detail on these mechanisms at this point in the presentation because each of them are clearly exemplified in our portfolio choices, and I will come back to these features in that section. I'd like to turn now to the second component of our platform, which is our novel cell-targeted LNP delivery system, which enables biologically driven cell and tissue targeting. We are clearly building on the achievements of first and second generation LNP delivery technology with our approach. These generations of LNP systems have progressively added biodegradable lipid components and expanded the application space from the liver to include delivery to immune cells for the purpose of vaccine development. Against this background, we've sought to actively target our LNP system to specific cells and tissues on the basis of a highly specific engagement between a ligand on the surface of our LNP and its cognate receptor expressed in a specific cell type or tissue. To accomplish this, we have first eliminated off-target distribution to the typical scavenger organs, such as spleen and lung. And then we have added back specific biodistribution to targeted cells and tissues through the selection of these cognate ligand receptor pairings. This results in two key features: improved tolerability and the ability to move beyond the liver and immune cells to address novel tissues both systemically and on a local basis. Today I'll review data on our progress to date in the liver, retina, and skeletal muscle, but we also have programs to address tumor and the central nervous system. For our ctLNP in the liver, we have focused on GalNAc as a ligand targeting the asialoglycoprotein receptor as its cognate. We like this receptor because it's highly and selectively expressed across species, which is demonstrated in the 4 photomicrographs on the left. You can see high density of the expression of this receptor across the species, from mouse to rat to non-human primate and to human. Secondly, this GalNAc and ASGPR pairing is highly conserved across non-human primates and humans, with a strong correlation in pharmacology between these two species. Importantly, ASGPR retains high expression in diseased liver, which is critical for programs that involve intrinsic liver disease, as we'll talk about in our portfolio. Our cell-targeted LNP delivery system provides highly selective biodistribution in mice, avoiding off-target delivery to the spleen. On the left side of this slide, you can see that our ctLNP delivers 97% of all copies of SEDNA to the liver. The sum of blood, lung, spleen, and kidney receive less than 3% of the total copies delivered. On the right side of the slide, this stands in contrast to first-generation LNPs, which demonstrate equal distribution of copies between the liver and the spleen. On the right side of that figure, you can see that we are roughly delivering 2,000-fold greater copies of SEDNA to the liver over that delivered to the spleen. This biodistribution profile enables dose-dependent expression in the liver in mice. Here we show doses of 0.5 and 2 milligrams per kilogram with proportional increase in expression at the left side of this graph. On the right, you can see that the luciferase reporter molecule is being expressed as expected solely from the liver. We have dosed up to 5 milligrams per kilogram in mice with good tolerability, and so far dosed up in non-human primate to 0.5 milligrams per kilogram, also with good tolerability. This profile enables our liver portfolio, which I'll turn to in a few slides. With the proof points we've observed in the liver for ctLNP, we moved on to examine this system in the retina. Of course, in the retina, we're not using GalNAc, but instead a unique composition tailored for this organ. At the top of this figure, we observe a high level of expression shown across 14 days, and critically, this comes with good tolerability shown in the optical tomography images below. We use imaging at 21 days post-dose and measure the outer nuclear layer thickness as the most rigorous measure of tolerability in this tissue. Here we see no thinning of the retina with ctLNP at 21 days. In contrast, second-generation lipid nanoparticles shows no expression and significant retinal thinning in the far right photo, our OCT image on the right. We've observed this thinning of the outer nuclear layer with all non-viral delivery systems we've looked at to date, and this profile for ctLNP is highly differentiated in our experience. Finally, I want to touch on some data that shows the progress we're making in moving beyond the liver and the retina into other tissues, in this case skeletal muscle. Here we've replaced GalNAc targeting ligand with a peptide that's specific for receptors on skeletal muscle cells. We've shown that this muscle-targeted CTLNP is selectively taken up efficiently in muscle cells in vitro, unlike the non-targeted control LNP. This difference in uptake in vitro is quantified in the bar graph on the bottom right. The next steps in this program are to continue to optimize ligand selection and conjugation and to move to in vivo systemic distribution and proof of concepts. Clearly, the ability to deliver full-length constructs to address diseases of the skeletal muscle with systemic redosability is of significant interest to us. So to summarize our CTLNP component of the platform, we've demonstrated the ability to eliminate accumulation of CTLNP in clearance organs and to selectively target liver systemically, which sets the foundation for our work in muscle and tumors. We've also demonstrated potency and tolerability in the retina, including expression in photoreceptors, which sets the foundation for extension into CNS applications. I'd like to turn now to the third component of our platform, which is our capsid-free manufacturing. I earlier mentioned the scalability and operating leverage of this process, and I'd like to step through the components here. Our manufacturing process is based on the SF9 suspension system. Unlike AAV, we do not bud virus, we do not create capsid, and this enables capacity scaling up to 20,000 liters, similar to that achieved by mammalian systems like CHO or HEC. We then harvest sedna by lysing the cells, which allows us to pair an equally scalable downstream column chromatography system. This pairing and scale between upstream and downstream is crucial to achieving both scale and operating leverage on this platform. This also yields exquisite control over purity and the analytical characteristics of the resulting products. We are routinely producing sedna at over 99% purity, and we show an example analytical chromatogram here at the 200-liter scale showing what this looks like, and we've achieved this robustly batch to batch. And while our CTLNP is highly differentiated biologically, the process for manufacture is very similar to that as for traditional LNPs, a flow system that is scalable to match the upstream and downstream process I've reviewed. Finally, we've had an opportunity to discuss these elements of the platform with the FDA during an Interact meeting conducted in December of last year. This is the current version of what used to be called the pre-IND meeting. We had a strong representation from the FDA, and we had the opportunity to walk them through our integrated non-viral gene therapy platform on the one hand, and our early thinking about the IND plan for PKU. We were able in the conversation to walk through these elements and to gain alignment around the characterization of our drug products, the analytical and manufacturing strategy, our GMP starting materials, and our non-clinical safety strategy. This allows us to proceed with clarity as we prepare ultimately for IND submissions in the future. I'd now like to outline our broad and expansive portfolio. And as we've established and selected these programs, we've prioritized those diseases with high unmet need and therapeutic opportunity, and specifically those in which the unique elements of the platform give us key differentiation from other approaches. Before going into the specific programs, I'd like to walk you through the rapid research cycle which has allowed us to develop this degree of breadth. We have a highly efficient research design cycle which enables us to quickly and efficiently generate new disease programs. It begins by designing constructs in silico and producing them in plasmid form, enabling in vitro activity screening and the production of selected closed-end DNA constructs for in vivo screening. This entire sequence from digital design to in vivo proof points can be accomplished in just 8 weeks. I'm going to begin by reviewing our program for PKU, which is one of our 2 most mature programs in the liver. Here we see initiating treatment in childhood to prevent neurocognitive decline as the key differentiator for this program. PKU is an inborn error of metabolism caused by a deficiency in phenylalanine hydroxylase, resulting in very high levels of phenylalanine in the blood that cause progressive, irreversible cognitive impairment beginning in infancy and progressing throughout childhood and adolescence. So the unmet need in PKU is clear: to completely normalize phenylalanine, we know exactly the level that we would need to achieve to enable normal cognitive development, which is less than 360 micromolar. We've generated a Sedna PAH construct and demonstrated that it expresses full-length wild-type PAH. Importantly, when we systemically administer these constructs into PKU mice that have very high baseline levels of phenylalanine in the blood, we see that all animals are completely corrected below the target level of 360 micromolar. We've learned a lot about PKU in the last 5 years in terms of what we need to shoot for, and I'll outline our approach on the next slide. Significant progress has been made in reducing phenylalanine levels with current standard of care, but it is not enough on its own to reduce the necessary levels of phenylalanine to below 360 micromolar, and this is shown on the left side of the figure. In the middle, diet doesn't either. Even strict adherence to protein-restricted diet results in spikes in phenylalanine up to 1200 micromolar, which is correlated with the irreversible decline in full-spectrum IQ. In the right panel, we see that adherence to diet diminishes as patients enter the second decade of life, which is a critical period for cognitive development, meaning that their phenylalanine levels are even higher and contributing to ongoing decline in neurocognitive function. Our goal is to achieve early, consistent control of phenylalanine levels to below 360 micromolar, and to do so by titrating the dose of our therapeutics to allow patients to live with a normal diet. We also have a program to develop a therapeutic for hemophilia A, where like in PKU, redosable childhood intervention and titration to the correct level for each patient is crucial to advance the standard of care for this disease. Hemophilia A is a well-known and relatively common rare disease in which patients have their first bleed in childhood. Genetic correction early in life is important to avoid the irreversible consequences of these early bleeds. This is an indication where the larger payload capacity of Sedna can be used to improve the potency of the constructs used in an eventual therapeutic. In the top panel of the slide, you can see 3 constructs which are derived from the sequences being used in development for AAV therapeutics today. The 4th construct is larger, significantly larger than the 4.7 kilobases, which represents the constraint for what can fit into the AAV capsid. This 4th construct, using more potent promoters and other regulatory elements, shows significantly increased potency when administered to mice at the same dose as is the Sedna construct containing the AAV sequences in development on the left. This greater potency matters because it enables a lower dose to achieve therapeutic levels of factor VIII, potentially reducing the burden on hepatocytes as they elaborate a systemic protein that is not normally produced by this cell type. How will we apply this increased potency in the context of the broader background of features that our platform offers to patients with hemophilia A? Well, on the left panel, we can see that in AAV gene therapy, all patients get the same dose. And in spite of that, there is significant variability in the observed factor VIII level across the patient cohort. Some patients are too high and at a thrombotic risk, and others are too low and at a persistent bleeding risk. You could not predict the outcome for each patient up front, and once it's in place, there's nothing you can do about it. And over time, expression tends to wane as you get out to 5 years. In our hemophilia program, we would seek to titrate the dose at the onset of therapy to get each patient in the correct range between 20 and 150% of wild-type factor VIII expression level by successfully offering additional doses as needed to get every patient in the right range with no patient overshooting. Of course, if patients were to wane, we would be able to offer the same therapy again. The right side of the slide explores the application of this approach by segmenting the existing population of factor VIII patients or hemophilia A patients by age and severity. The target population for existing AAV gene therapy is in the top right quadrant, adults with severe hemophilia A who are on prophylaxis. Even within this segment, we know many patients will not have an opportunity to benefit from AAV due to pre-existing antibodies, and still more will not achieve the optimal results due to the variability we discussed. We believe these patients will be eligible for our therapy in the future, and so will those whose effect is waning over time. But even more importantly, we believe we can uniquely offer dosing to pediatric patients with initiation of therapy early in life. In hemophilia A, as is the case in many genetic diseases, the preponderance of addressable patients are of pediatric age. We believe we will both expand access within existing therapy segments for AAV, but also open up very important new segments for patients with this disease. I'd like to describe our program now for Wilson disease. This is an example of a program designed to deliver a large gene to prevent liver transplant in childhood. Wilson disease is a progressively fibrotic condition of the liver that results in end-stage liver disease requiring liver transplantation, often by the second decade of life. The disease is caused by a deficiency of the gene ATP7B, which is a gene too large to deliver in its entirety within the constraints of AAV. In our Wilson program, we've produced full-length ATP7B, as shown in the immunoblot. And on the right, we have demonstrated that full-length protein expresses within the cells and in the appropriate subcellular locale around the nucleus in the Golgi apparatus. We see an advantage to administering the full-length protein, which contains the full complement of copper-binding domains, to alter the progression of the disease and ultimately avoid the need for transplantation. Gaucher disease is a common lysosomal storage disease impacting about 6,000 patients in the U.S. who are typically diagnosed in late childhood or adolescence. It results from a deficiency of GCase enzyme, and the current standard of care involves weekly or every other weekly enzyme replacement therapy. In our program, we've shown in vivo that one-time IV delivery of sedna containing the sequences for GCase result in therapeutic levels of GCase expression from the liver into the blood. The key here is that constitutive expression, constant expression of the enzyme at the same level over time, may be very different and offer advantages over an every week or every other week delivery of enzyme replacement therapy, because the activity of the enzyme is only active for a short period of time, whereas constitutive expression may be more effective in reaching the key tissues and removing the toxic metabolites that accumulate in liver, spleen, and bone. Clearly, if we could achieve those benefits with an every 5-year dosing regimen, it would represent a significant reduction in the burden of therapy for patients. Finally, in our liver portfolio, I'd like to turn to antibody gene therapy, where we seek to deliver antibody genes to express and secrete monoclonal antibodies from the liver. There are therapeutically relevant antibodies for multiple infectious diseases, including HIV, RSV, hepatitis B, and we've previously announced our collaboration with Vir to deliver the sequence for potentially neutralizing antibodies against CoV-2. The goal of antibody gene therapy is to enable constitutive expression above 1 to 5 micrograms per mil in the body, expressed from the liver. Passive administration of previously produced or exogenously produced monoclonal antibodies cannot do that over long periods of time, with on average 3 to 6 months of efficacy per dose. In our program, we've administered our sedna construct intravenously into animals, and we see expression from the liver of over 40 micrograms per mil after a single administration. We believe that by combining durable immunity up to 5 years per dose with the cost effectiveness and scale of our platform provides the potential for globally relevant treatment of infectious diseases. Furthermore, we believe the future of antibody gene therapy more generally lies in their expression and regulation from the nucleus of patients' own cells. I'd like to turn now to our 3 programs in the retina, beginning with Leber's congenital amaurosis 10, or LCA10. Here, the advantage of the platform is to deliver the full gene to address all patients with a single therapy. LCA10 is a disease usually diagnosed at birth with poor or absent vision due to a mutation in the CEP290 gene, which is too large to fit within the constraints of the AAV capsid. We have produced and expressed full-length CEP290 as seen in the immunoblot in the top middle part of the slide. One of the advantages of LCA10 in the clinic is that it's possible to demonstrate restoration of vision on a relatively short time frame. So it's an appropriate candidate for both a rapid and robust human proof of concept in the tissue. We also view the fact that we can replace function of the entire gene as highly differentiating because we can potentially offer therapy to all LCA10 patients, irrespective of their underlying mutation. Moving on to Stargardt disease, we see here a program where we seek to deliver a large gene with native regulation to the right cell types, characteristics that are unique and important to changing the course of this highly prevalent ocular disease. The reason that the combination of native regulation with the ABCA4 gene is so important is that to alter the course of Stargardt disease, it's key to deliver appropriate levels of ABCA4 to photoreceptors and lower those still significant levels to cells of the retinal pigmented epithelium. And it is equally important not to deliver significant levels of ABCA4 expression in other retinal cell types. We believe that the combination of the native promoter, which normally regulates and sets the level of expression according to the local environment and cell type, is the best and most efficient way to achieve this outcome. We have produced and expressed full-length ABCA4 in combination with its native promoter, and when we deliver that construct to cells, we observe that ABCA4 is expressed within the cells in the membrane fraction, which is exactly where you would need it to be to support appropriate function. Finally, in the retinal portfolio, we turn to wet age-related macular degeneration, where we seek to offer a very long-lasting and ultimately redosable gene therapy for this condition. This is obviously a highly prevalent disease with really excellent existing standard of care, which is mainly limited by the frequency of therapy that's required to control the disease. So it's a well-known target and a well-established market, and our wet AMD program seeks to deliver Sedna using cell-targeted LNP to constitutively express a VEGF-binding monoclonal antibody in the retina. In the middle graph, we've utilized our ctLNP to deliver intravitreally, in this case using mRNA as a reporter modality, and we see robust retinal expression of the transgene across a variety of key cell types. Our next step here is to optimize our delivery system to enable intravitreal delivery of closed-end DNA, offering a very long-lived single-dose therapy that ultimately could be redosed either to achieve titration or to extend benefit. Finally, I mentioned that we are working on 3 areas of expansion beyond the existing portfolio of clinical programs. Here we seek to change the targeting ligand for the cell-targeted LNP in order to deliver Sedna therapeutics to new tissues. We're particularly excited, as I mentioned, based on our early in vitro data in the skeletal muscle, to develop therapeutics that could deliver full-length skeletal muscle genes such as dystrophin for Duchenne's muscular dystrophy or sarcoglycan for limb-girdle muscular dystrophy. For these diseases in particular, the additional component of redosing could be an important aspect to ensure broad, repeatable biodistribution to a relatively large tissue compartment. Our interest in tumors derives from the opportunity to select tumor-specific ligands to systemically direct Sedna therapeutics that could localize to tumors, delivering payloads to include cytokines, antibodies, or other proteins in order to change the course of tumor growth and or distribution. And finally, in the central nervous system, we similarly seek to swap out CNS-specific ligands to drive expression of specific genes to influence genetic diseases of the central nervous system, such as Dravet syndrome or Hunter syndrome or MPS II. We are building a major biotech innovator here at Generation Bio on the basis of what I've shared today. I'm going to turn now to what lies ahead for the company. Now I'd like to outline the anticipated milestones that are at the core of our work over the next 2.5 years. Starting with our liver portfolio, the balance of this year will focus on demonstrating disease proof of concept in the animal models for PKU and hemophilia A. Here we'll be pairing our ctLNP system with our lead constructs, showing disease control and titration in each case. We'll also be working to demonstrate NHP expression with the ctLNP system using factor IX as a reporter construct. This work in the balance of 2020 in the liver will lead to IND-enabling studies for PKU and hemophilia next year, as well as lead optimization in the additional diseases that are deeper in the liver portfolio, ultimately culminating in the liver with our first IND submissions for PKU and hemophilia A in 2022, and deeper preclinical development in these additional liver indications. The balance of 2020 in the retina will be focused on showing the delivery using the ctLNP system in that tissue to achieve in vivo proof of concept points in the disease models for LCA10, Stargardt, and wet AMD. Building on this momentum in 2021 in the retina will be lead optimization for our lead program in that tissue, as well as demonstrating NHP expression. Ultimately turning towards preclinical development for our lead program in the retina and additional proof of concepts in our existing portfolio. Our manufacturing platform will continue to operate at 200 liters for the balance of this year, supporting our existing contract manufacturing organization as well as our ongoing tech transfer to our second provider. 2021 and 2022 will be focused on scaling both the upstream and the downstream for our cGMP supply chain. Next year, we seek to establish ctLNP delivery and proof of concept in vivo in our expansion tissues of skeletal muscle, oncology, and CNS, leading in turn to lead optimization in these tissues in 2022. In creating this new class of non-viral gene therapy, we aim to use our platform to rapidly extend the scope and scale of gene therapy, to access more patients in existing indications, to reach new diseases, and to bring gene therapy onto the global stage as a new pillar of medicine. Thank you for your time. It's been great to be able to share with you our ambition and progress here at Generation Bio, and hope to engage with you in the coming IPO process. Thank you.